Hey, good morning. I hope everyone are done with our assignments and you have done with watching the videos. So far, we have seen how to use a DNA sequence to find its identity through blast analysis, how to construct a phylogram using the DNA sequence and reference sequences taken from the NCBI database, how to study the pairwise distance data, and how to prepare the Excel sheet using colored scale bars. Okay, I hope everyone is clear till now. And before we start our own project, which is today, I want you guys to practice this BioEdit. Okay, what is this BioEdit? Last class, we have seen how to extract the DNA sequence from the chromatogram. Okay, as you have seen, we are actually doing two-way sequencing. What is this two-way sequencing? We are going to sequence both the ways. Why to DNA occurs in double standard DNA, and we are going to sequence both forward and reverse strand. Why? Okay, even people who are working in, say, for example, 500 base per gene, they will again go for two-way sequencing. As I told you in last class, if it is 1500 base pair, sequencing both is very much essential to obtain the completeness of the entire length. Okay, as in the modern day DNA sequencer, they may produce up to 900 or 1000 base pair. So if you want to cover 1500, better sequence both the strands from both the ways. Okay, when I say both the ways, usually the DNA strands are read from 5 prime to 3 prime. Okay, so by now you should be very clear that in molecular biology, when you say DNA extraction, you are extracting the entire genome, which contains thousands of genes. Okay. How we particularly amplify our own desired gene or our target is by using primer during the PCR. Okay. This is a primer sequence in the picture, where the forward primer bind to a specific region and the reverse primer complement to the particular gene, and they will just amplify the particular fragment. Okay, so using primer, that's like baiting. You are getting what you want. Which gene to amplify is decided only in PCR. Okay. Even people working with 500 base per gene, they will always sequence both the strands, because sequencing two ways also ensures accuracy. Okay. You see, if this primer is committing a mistake, when I say mistake, it means instead of thymine, it will be adenine here. Instead of adenine, if it is adding guanine, you can verify this position by comparing with your reverse complement. What is this reverse complement? Let's now understand just the reverse sequence. When you have a reverse sequence, it acts as a one more layer to check whether the forward sequence is correct or not, and it is true vice versa. Okay. So for error correction, accuracy, completeness, you need to go for two-way sequencing. Meaning, when you are done with your PCR, the PCR amplicons are split into two samples, and they are again subjected to single strand amplification. Okay, so single strand, either forward strand or reverse strand, is amplified separately. Again, both the samples are injected into the sequencer, ABA sequencer. So you should know, when you submit a sample to the company, they actually isolate DNA, they go for PCR, then if necessary, they will do PCR purification, then they will go for single standard PCR, where your sample is split into two single strands. In one PCR tube, only forward primer is added. In another PCR tube, only reverse is added. That's how the single strand amplification is facilitated. And that amplicon is subjected to two sequencing reactions. So every sample in the sequencer, when they are loading, it will be two samples, two single standard PCRs. So for every sample, you will be getting two machine output files. Okay. Now this BioEdit is it's entirely about how to use these two strands, how to compile them and obtain our target DNA, complete length of target DNA, or how to check our accuracy of the DNA sequence and everything. Okay. So I will, to understand, I hope this picture is very clear. One strand is a forward strand, other is reverse strand. So you will be having two machine output files. Okay, for analysis. So before we proceed, I just want to recall again, what is this reverse complement? Okay, see here, say for example, consider this 10 base pair DNA strand. Okay, A, triple C, double G, T. Okay, this strand. I hope you know this complementation. Adenine always pair with thymine, adenine, thymine, adenine, thymine, cytosine, guanine, cytosine, guanine, cytosine, guanine, guanine, cytosine, guanine, cytosine, thymine, adenine. Okay. So adenine always pairs with thymine and the cytosine always pairs with guanine in triple hydrogen bond. Adenine and thymine is always double hydrogen bond. You know that. Okay. So what is this reverse complement? Reverse complement is nothing but if I am reading this is a forward sequence A, C, 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 G, G, T. Okay. And the reverse sequence we are seeing this. Reverse complement is something you have to read the sequence in reverse and also in complemented manner. Okay. So what is this? Okay. See here, I'm going to read the sequence in reverse. Reading the sequence of this sequence in reverse is T, G, G, triple C, WA. Okay. Instead of reading like this, instead of T, I'm putting A here. Instead of G, C. I'm complementing the reverse. That is called reverse complement. So T, A for G, C, here G, C, triple C, triple G, WA, double T, double T. Okay. It's all about ATGC, all four nucleotide. This is reverse complement. Why to do this reverse complement? You will know. Okay. Only by reverse complementing the reverse strand, you are actually merging or you are actually completing your forward strand. I will tell you how it is done, or I will show you how it is done. Okay. So I hope this is very clear now. Okay. DNA are double stranded. Okay. So positive strand, the forward strand is something like this. In another example, just to make my point. Okay. AGTCAGTAACTAG. This is the forward strand, and this is reverse complement to the forward strand. Read it from the reverse. GC. A complement is T. T complement is A. C complement is G. So this is reverse complement of the forward strand. Okay. I hope this is very clear now. So how to merge the two machine output file? It is a well-known fact now that you have to dedicatedly sit and edit each and every base pair in the machine output file. After doing that, you can extract the sequence of forward and reverse and you can do the merging. How to do this merging? That's when the BioEdit comes into play. Okay. So these are the 8 important steps you need to follow to merge the DNA sequence. Instead of reading each and every step, I know it will go to freak you out. Instead of reading, I'm going to show you how to merge the sequence. Okay. So what I will do is, I will take an example file. Here, uh, I have a client's sequencing file here. Okay. So I edited each forward sequence and reverse sequence. I copied those edited sequences in the notepad file. Okay. So here I have a forward sequence. Okay. Here I have a reverse sequence. I always believe Word document is an excellent um tool for DNA compilation and counting number of base pair. So I am going to use my Word file to show you how it is done. Okay. So I'm opening my Word file. First, you copy both the forward and reverse sequence in the notepad file to Word file. Okay. So I have copied. This is my forward machine output file. This is my reverse machine output file. I have edited each and every chromatogram. I got this. Okay. So the first step is, as you have copy and paste the forward and reverse sequence into the Word file. Done. Open BioEdit and reverse complement the reverse sequence. What it is, I will tell you. So before opening BioEdit, you should know how to download the BioEdit. So I'm showing you. In a Google, just type BioEdit. Okay. The BioEdit download, bioedit.software.informer.com. This is an official website. Download, go here. Okay. This first option, download the latest development. This is the latest file. Okay. 15 MB. Just click it. Now download is in progress. Okay. I can see it here. Once downloaded or download is done, they will appear as a zip file. Okay, there are multiple files downloaded, maybe because I clicked multiple times, but just unzip the file. Extract all. So extraction is done. The file is opening. Okay. It will be as if file double clicking the setup, you can easily install the, okay. Once the installation is successful, you will see a okay. You can check the availability of so this is because I already have BioEdit installed. Okay, so just for demonstration, I showed you how to install the BioEdit. Once the installation is done, you will see something like this. A window something will opens like this. Okay, so this is BioEdit. And what is been the second step? Open the BioEdit. I have opened it. Reverse complement the reverse sequence, meaning copy the reverse sequence alone and reverse complement it. You know, if it is a 10 base pair, I can sit here and I can reverse complement the sequence, but it is around 600 base pair or 700 base pair here. So how to reverse complement using BioEdit? Just go to File, New from the clipboard. Since I have copied the sequence already, I have copied, right? Copy. Then it is in the clipboard of the computer now. From the clipboard, New from the clipboard. If I click this, now the sequence is being imported into the BioEdit. Okay, I will click the sequence title. I'm going to reverse complement the sequence. I will increase a font for you to see how the reverse complement is done. See sequence, nucleic acid. The third option I clicked, third option sequence, come down, nucleic acids. In nucleic acids, reverse complement. I click it, I'm going to click it, just observe the text here, they will be complemented immediately, right? 2, 3, 1. Can you see that reverse complemented? Now the entire sequence is reverse complemented. Now I want to copy this sequence again, paste it in the Word. So go to the second option, edit, copy sequences to the clipboard. Then I will open the file and pasting it here. Yes, this is the reverse complement of the reverse sequence. See, it starts from CC, but this is TGG. If you see here, this T actually TCCG actually represents to AGGC. Can you see that the entire sequence has been reverse complemented here? Now I don't need the reverse sequence. I'm deleting it. I have a forward sequence. I have a reverse complement of the reverse sequence. I would say reverse complements are extension of the forward sequence. How? Just copy the last fragment, 10 base pair. Okay, around 10 base pair, I'm copying it here. In the Word document, Control F, find, I'm pasting it here. See? So this means the sequence ends by CAAG, CAAG. After this is an extension. So I don't need these portions now. Okay, I'm going to merge it. That's it. Okay, that's how the forward and reverse sequence are merged. See, this is possible only if you have meticulously prepared the chromatogram. If you have left one, two base pair differences, we'll be in trouble. Okay, this is we can also merge this in Mega, and we can also merge and prepare a consensus sequences in BioEdit. But I prefer to do it in Word because many more things can be done. Now I am checking the well length. It is 1470 base pair, which is more than 90% length or 85% length of the 6S RNA gene. Okay, so that's how the merging is done. That's what the instruction is also about. Once delete the original file. Okay, from the forward sequence, copy 10 base pair. Use Control F to find this 10 base pair, the reverse complement. Once match, copy the remaining part of the reverse complement after the 10th base pair overlap. Append it to the end of the. So that's how the merging is done. Okay. So today it's a new tool, BioEdit, because we want to compile both forward and reverse chromatograms by reverse complementing it. Okay. These are the steps for reverse complementing. I hope you understand this. Today I will be sending you guys the forward and reverse extracted sequences. So you reverse complement that and you go for blast analysis. Let me know how this blast results turns out. Okay. So today again we will fix on our individual topic. I will share one more video regarding that. Thank you.